Hello and welcome. I'm Kate Woda, Director of the Immuno-oncology 360 Summit, or IO360 for short. Today's fireside chat explores the future of CAR-T, cell therapy, T-cell engagers, and synthetic immunity. We'll discuss the contrasts in functionality and prospects of CAR-T and T-cell engagers in single and combination therapies, shedding light on their unique features in cancer treatment, especially for solid tumors. After the discussion between Dr. June and Dr. Chen, there'll be a live Q&A. Please submit your questions in the chat feature through the webinar, and we will address as many as possible. Please make sure that your chat is set to everyone as the default. Now, before we proceed further, I'm thrilled to share some exciting news. Mark your calendars for the upcoming 10th annual IO360 Summit, taking place February 27th through 29th in New York City. And today's discussion is just a preview of the innovative topics and insights awaiting us at the summit. So without further ado, I'd like to welcome our guests, Dr. Daniel Chen, founder of Ingenuity Life Sciences and Synthetic Design Lab, with Dr. Carl June, director of the Center for Cellular Immunotherapies at the Perelman School of Medicine at the University of Pennsylvania. Carl and Dan, welcome. Okay. So now I'd like to turn it over to you, Dr. Chen, to get us started.
Wonderful. Well, thanks so much, Kate, to you and IO360 for putting this on. How you doing, Carl? You know, it's great to see your face across the country. And it's hard not to, when I look at you, not be reminded of that moment that you and I shared with IO360 back in February of 2020. That was a debate for you and I on CAR T cells and T cell engagers. It felt like a really important watershed moment. Charlie Graber was our moderator for that. And it was a really important time in the field of cancer immunotherapy and synthetic immunity. However, I think most of us didn't quite recognize, perhaps you did, how big a moment it was going to be globally, because we were sitting right at what would be the beginning of the pandemic, at least for the West. And you showed up to that debate wearing a mask, and you did the whole debate wearing a mask. And it was so shocking. The world changed after that. And I felt like you sent a big message to everyone that was there and saw that. So your thoughts on where we were back then versus where we are now?
Thanks, Dan. First of all, it's great to be back. And I think at a really appropriate time because, we were just going into a COVID pandemic and now we're really completely coming out of it, I think. So, we can look back in hindsight at where we were. And we now have more CAR T cells approved than we did. We have many more kinds of bispecific T cells engagers, approved. And what we found out is that the market has nowhere near plateaued. It's just the scope of this is really increasing, and in spite of what happened during the lockdown.
Yeah, so I mean, it does feel like we learned a lot, not only about our world during that period of time, but I think COVID actually led to an amazing advancement in science. A lot of it was infectious disease related, but a lot of it was actually just immunology related. And I think we learned a lot about cancer immunotherapy and immunology coming out of all of that. But for us today, here we are, we're back talking about CAR T cells, cell therapy, T cell engagers. It doesn't feel quite fair because you and I did this previously as a debate, and now I get to be the moderator. But I think this is an opportunity for you and I to sort of revisit where we are and hopefully really push each other on where we think we stand with synthetic immunity. There's been amazing advances. We've learned a lot, but there have also been a lot of challenges that seem to have popped up during that period.
Yeah, I mean, there's been, I think one way to classify things would be what we have had emerge on the scientific learnings. And then the other part is the engineering of scaling out this technology. And there have been challenges in both. I think, we've seen issues both with toxicities, and we now know, which we didn't know back when you and I talked then, that the durability of some of these therapies now is out past a decade.
Yeah.
So it's helped realize the promise of what a cell and gene therapy can do and what synthetic immunity can bring. But we have to understand the delivery of these as a challenge that the biopharma has never faced. It's a huge challenge.
Well, so since you bring some of those challenges up, FDA is making comments yesterday and today on the field of cell therapy. I think there's some confusion around what that really means for the field. Is it impactful or not? What is really happening there? I don't think anyone is better qualified to comment on that than yourself.
Well, thanks, Dan. I think so. What happened was on November 28th, the FDA issued a statement that they were investigating T-cell lymphoma cases that have been reported to a voluntary database that the FDA maintains, and that they were investigating the causes of these. And then, as you just mentioned, today they have now sent letters to each of the 6 commercially approved CAR-T cell therapies, and this is for lymphoma, leukemia, and myeloma, and asked that they revise their warning to patients and physicians. And that warning in specific is that T-cell lymphomas have been reported in patients after treatment, and the database that the FDA said back in late November was on 34,000 patients infused, with the commercially approved CAR-T cells. And what the FDA has also said is, they're investigating the mechanisms here, and that the risk-benefit ratio still remains favorable. And we have to put this in perspective. We've known for at least 30 years that chemotherapy and radiation cause tumors, and we have a lot of molecular explanations on how that happens. But we don't have a really good prospective database to know how often it happens in patients.
Right.
And the beauty of this here, in the case of cell and gene therapy, is that the FDA has a prospective database where manufacturers are required to report adverse events and to track patients for 15 years. So we have now a numerator and a denominator. So 28 cases over 34,000. Turns out very few of these cases of T-cell lymphomas can it look like it's really due to the CAR transgene that's in the T-cell, meaning when the lymphomas are sequenced and to the level of certainty that we have so far, the transgene's not in there, meaning that it came from the host, and that most likely the patient has gotten a secondary tumor over whatever their initial tumor was, myeloma or a leukemia or lymphoma.
Well, it seems like the good thing is that the clinical benefit for cell therapy is very high, and these events seem to be rare, but not fully understood yet. So it seems good that we will track that over time to better understand it. One thing that I think has expanded in scope since the last time we talked is the emergence of more activity in related TCR fields. So TCR-Ts and maybe TCR engagers. Your thoughts on these versus CAR-T and tumor-associated antigen-directed T-cell engagers?
Sure, we don't yet have commercially approved TCR-Ts. You have to remember, though, we have been treating patients with TCR-Ts that are not genetically engineered, for more than 40 years. And that would be the TIL therapy that initially Steve Rosenberg and groups have done in patients with many cancers, but most prominently melanoma. And even with unengineered cells, there have been responses that have been durable. And there's extensive, preclinical data in mice that T-cell receptors have the possibility to recognize either 2 kinds of antigens: shared antigens, such as a mutated KRAS, which is the most common oncogene and you can target that with T-cell receptors, or so-called neoantigens, which are those that occur through DNA mutations in patients, and then And that's opening up a new kind of personalized therapy with TCR-T. So over this last year, the data continues to mature, and it looks like the first approvals will be in solid tumors with HLA restricted TCR-Ts and autologous T cells, and likely in solid tumors such as certain forms of sarcoma.
Yeah, I mean, I think that you're absolutely right in that the TCR-based therapies allow us to expand, importantly, the two types of tumor antigens we can go after. Obviously, they're a little more complicated in the sense of, all of the HLA selection that has to go on. But it feels like one of the other potential advantages for some of these is possibly more physiologic stimulation and maybe bridging what cell therapy currently looks like with what endogenous immune approaches look like. Your thoughts on that?
Well, exactly. I think, for instance, in some cases, we're looking at boosting what would be natural immunity and adding in new specificities. And in that case, probably the best thing is what Darwin evolved, which is a highly tuned T cell that we all have, and now we can redirect its specificity. But I think on the other side is the aspect of synthetic immunity, which we're discussing today, which is we have amazing ways now to, in the cases of where we have disturbed immunity, such as a tumor where it makes immunosuppression, or HIV, where the same kind of thing can happen with genetic editing, and that can use things from CRISPRs to zinc finger nucleases. Base editing is a huge new approach here. And then adding in other molecules that all can make a cell resistant to what would otherwise shut it down. So I think we are going to see a very large expansion of this area of TCR T cells, and it will be both natural T cells and synthetically enhanced cells.
Mm-hmm. So we have lots of time to spend on what I think are incredibly exciting advances in the field, but maybe let's turn for a moment to perhaps one of the things that those that are more peripheral to this field maybe found disappointing over the last couple of years, and that was probably the difficulty that solid tumors have presented for synthetic immunity. I think many people, if you ask them, I don't know, 4 years ago, 3 years ago, 5 years ago, they would have thought, oh, we were able to go from 0 to 60 in lymphoma and myeloma and just completely change clinical outcomes for patients with synthetic immunity, what felt like overnight. And I think many people felt like this is just going to sweep through all of cancer and completely revolutionize solid tumor treatment. But that hasn't quite happened. Your thoughts on what has made solid tumors such a difficult challenge for synthetic immunity?
Sure. I think we have to remember the history that TIL therapy basically only worked with any kind of reproducibility in melanoma. And we now know, with all the sequencing, that melanoma is one of the most extreme examples of what we would call a hot tumor, where in the baseline, the immune system has found the enemy, and when you biopsy the tumor, you find infiltrating T cells. And that's where checkpoint therapy has worked the best. But when you go to the predominance of solid tumors like pancreatic cancer and most sarcomas, they are extreme versions of cold tumors, meaning that when you biopsy them, they're an immune desert, as you described many years ago with Ira Melman. So these immune deserts, there's no T cells there in the first place. And in many cases, the immune system has either been completely shut down or it's never even known that there's a tumor there. So that is where I think we have the biggest room to gain with synthetic immunity and expensive cell therapies, because there are huge issues with T cells inducing, the tumor inducing T cell dysfunction, exhaustion, cell death, and there's barriers to even getting into the tumor, such as most notably pancreatic cancer, where often there's a stroma with collagen-rich areas, which make it impossible for natural T cells to enter into the tumor. So this issue is the one main one with this T cell dysfunction and a cold tumor. The other is that we were in a sense lucky with myeloma and blood cancer because we had lineage specific antigens that you can live without. So in the case of leukemia CD19, there had been genetic, cases characterized in the pediatric literature with people born without CD19, and yet the kids were fine as long as they got antibody replacement therapy. And we now know that CD19 deficiency, meaning B cell aplasia, in adults is very well tolerated. That was actually a surprise, but that's turned out to be true. And we also had in myeloma several lineage-specific antigens, most notably BCMA, which turns out that we can target those and make B cell aplasia, myeloma, plasma cell aplasia, and have very potent tumor eradication, but without any significant toxicities other than cytokine release syndrome and inflammation. The problem with solid tumors is, with very few exceptions, those dream targets don't exist.
Well, so I think you bookended it really nice in terms of the heme malignancies and the opportunity in the real immune deserts or immune excluded, where we don't see a lot of endogenous immunity. But I do think that there's a gray area in the middle that is really important for us to think about as we build better therapies in synthetic immunity. And that's all the tumors that checkpoint inhibitors and endogenous immunity have revolutionized. It's not just melanoma, right? It's lung cancer is completely different now that we have checkpoint inhibitors. Kidney cancer, bladder cancer, hepatocellular. So there's a pretty nice list of tumor types that sit sort of in the middle that have responded really well. And I think I certainly have found it interesting that we haven't been able to just immediately break into those spaces, which to me at least tells me that there is something fundamentally different about the synthetic immune response that we're creating that is different than the endogenous immune response. So I'd love to hear what you think in terms of what's driving that.
Yeah, well, I mean, that's exactly the problem, that there's basically been a glass ceiling of 20 to 30%. I mean, lung cancer, as you said, is totally revolutionized. And 20 or 30% of the patients now can get 5-year survivals, which is amazing. When we trained as physicians a long time ago, that was never the case with metastatic lung cancer. So we've had a revolution. But, there's a lot of people that are being left behind on this. The majority still die of progressive disease. And this is where I think synthetic immunity is poised to overcome those challenges, which are, issues of the cell tumor-induced immune suppression, which there's a lot of research on this. And I think, in mice, we see a lot of progress. It still needs to happen in humans, though.
Yeah, I mean, when I look at it, it's certainly there's a plethora of things that it could be. You mentioned targets. It could be part of the tumor microenvironment that's suppressive. It could be metabolic. And in fact, a lot of these things probably all play into it. But to me, one of the ones that I'm most intrigued by is this question of whether with synthetic immunity, we're sort of overdoing it. We're getting too big a initial pulse of activity rather than what we see with endogenous immunity, which is sort of more built up and more moderated in terms of that stimulation of T cells. And so I'm curious as to how you view a few of the interesting data points coming in from cell therapy and T cell engagers. So I'll raise on one end, the excellent work that Crystal Mackel's group at Stanford really started on looking at tonic signaling and how that really can affect the function of these cells. And then the second one that I think is an interesting counterpoint to that is in the T cell engager space. the use of TKIs actually started again from cell therapy studies, looking at moderation of that signal and the ability to either turn it down or turn it off for periods of time, and how that affects the functional states of these cells, which do appear to be perhaps epigenetically driven. And so would, again, love to hear what you think. Like, do you think that this is a big part of the answer or not?
Well, I think it's one big part. Unfortunately, I don't think there's going to be one magic bullet here. And so I think there's a cell intrinsic modifications that will either reverse or prevent some of this epigenetic issues that cause the T cell dysfunction, where they become basically useless. And this can happen very early. There was an amazing report earlier this year that at SITC, where within 6 hours after a T cell entering a solid tumor microenvironment, the cells are rendered dysfunctional. So and so that's an acknowledged big challenge to the field. Now we found one way to delay that, at least, is by studying patients who've already been treated with CAR-T cells, and then look at the cells. If you look at, we may give 1,000 different kinds of integrated autologous T cells, and then look at the ones who perform the longest versus those who wear out right away. And then what we find is that the ones who continue to work longest have epigenetic changes. Like the most initial striking case we have found, and we've now found many of them, is patients who have, through the cell manufacturing process, a loss of the gene TET2, which it oxidizes methylcytosine on DNA, and that retards differentiation of the cells, and it makes them more resistant to exhaustion. And there have been now dozens and dozens of these kinds of things found. Sub 39, SUV 39H was just reported by Michelle Sadelain this last week as another one, and it's related to the TET2 family of epigenetic editors. So that's one approach, the cell intrinsic. But then I think there's going to be others required, which will deal with other aspects, such as direct checkpoint modification that we haven't yet addressed with checkpoints, and can be done in the T cell itself, or by giving other molecules, such as recombinant antibodies, in addition to the cell therapy. So anyway, I think it's both cell intrinsics issues and these other aspects that are cell extrinsic to the tumor microenvironment, trafficking and getting into the tumor, that need to be solved.
Well, I think these are exactly the kind of things that you can do with cell therapy that there's probably no other way to really effectively do. And so if you can change T cells in a way that allows them to preserve their functional state, even through the difficulties of getting into a solid tumor, and provide that prolonged anti-tumor activity, that's going to be a game changer for this field. And so one of the questions I have for you is just, how do you do that? Are you going to engineer in all these changes to control their functional state? Or is it just your ability, or can you do it actually as part of the signals you give in manufacturing or even in vivo? How do you look at that?
Well, I think, first of all, we need to do the trials to understand. I mean, because one approach is, can you do it an all-in-one like cell, and that you give it, and then it makes smart decisions when it gets in the right environment and knows when to shut itself off or turn itself on. That would be the end goal, I think, is to have one cell like that does that. But another way to do that is to have physicians give drugs, like you mentioned. What was shown at Stanford is giving like a drug like dasatinib to make the cell shut off, so the T cell stops signaling, and then by resting, it actually can recover its function, or at least part of it, and then you can remove the brakes, and then it takes off again. And so there are a number of approaches like that, of combinatorial therapies. And another way to do it would be to give not just one kind of cell, but to manufacture several. I mean, we've just had in Nature Medicine last week a really nice, the phase 2 results from MD Anderson of CAR-NK cells. So giving natural killer cells, who they kill in a different way. than alpha beta T cells do. And there are data that we could either get synergy by giving cells that are different subsets. So natural killer cell plus T cells, CAR macrophages plus T cells or NK cells, et cetera. So I think we're going to see all those fronts be tested.
Yeah, and you know that's an area that I really love. And back in 2020, I think I referred to it as, are you trying to, create a horse that can make its own decisions on where to go and what logs to jump over versus riding a bicycle where you maintain that control. And I think in the long run, if you can build it all into one cell that can do everything, that's fantastic. But it does feel like we're a long ways away from that. And being able to meet in the middle where you build the best cell you can with as much logic control that you can to adjust to its environment appropriately, but still allow a physician and a patient to be able to modify that response in their bodies by giving just different therapies. And so that feels like a very powerful way to go. You've also reported on some other interesting advances that I think are particularly intriguing. I think one of the ones that you've shown is the ability to add just a simple cytokine like IL-18 to CAR production and really change that portion of therapy. What do you think about that? Where do you think that's going, and how do you view that in terms of both manufacturing, but also is that the kind of thing that should be happening in vivo in the patients?
Yeah, well, totally. I think what we're seeing is one approach is gene subtraction, using ways to either knock out genes or to turn them off. And then like TET2 is one example. And there are many others. When you turn them off, you get prolonged activity with the cell. And then the other way is really gene additions. And you can, we have the technologies with viral vectors, with things like Sleeping Beauty, and to insert large chunks of DNA into cells, and sites specifically do that, in fact, now, rather than in a random way with many viral vectors. But with gene additions, then recombinant cytokines can be secreted, and other enzymes can be secreted, and many aspects of that can promote the activity of T cells or their trafficking, even chemokines and so on, that would direct them into the tumor metastasis and away from other areas of healthy tissue. So what we've done in an ongoing phase 1 trial is to take a CAR-T cell and then add in human IL-18, which is like IL-1. It gets secreted, with the inflammasome, and activates very strongly innate immunity, so macrophages and NK cells, but there are IL-18 receptors on T cells, so it can backbind and increase the activity of the T cell, but without having systemic toxicity. So this looks very promising in the ongoing trial.
Yeah, and IL-18 is an interesting one. When we first were developing Tecentriq in the phase 1, in that original Nature paper describing the phase 1 biomarker work, IL-18 was one of the ones that came out. It was one of very few things that pulled out, differentiating the type of clinical activity that was being seen. And so, it's interesting to think about that role and whether is it really going to exert the majority of its effect on T cells directly, as you talk about, versus change the community of innate immunity around those T cells?
Yeah, so that's like the $64,000 question. We don't have data yet to know. I think it's probably doing both. And I think a lot of people overlooked IL-18 because IL-12 seemed more potent in a lot of the preclinical models. And in fact, there's been a lot of toxicity with IL-12 over the years when it's been given either as recombinant cytokine or, genetically. And it turns out IL-18 has its own endogenous checkpoint called IL-18 binding protein. And this may build in safety over what we've seen previously with IL-12.
Yeah, so we'll certainly follow that closely, but you're right. IL-12 was so powerful in its biologic effect, but perhaps it had too broad an effect and has not been well tolerated in patients. So it limits, I guess, its therapeutic index is really what's happening there. Let's move on to some of the stuff that's really behind the scenes that a lot of people might not fully recognize. There's so much technology and engineering coming, and in many ways, cell therapy, but also protein engineering, in terms of therapeutics is, I mean, we've seen that explode since the original CARs were developed. And some of the stuff that I think has been most exciting has been started with the logic gating that folks like Wendell Lim and Cole Roybal developed at UCSF, in terms of starting to give the types of if then or, if then but type of logic that, you know, those of us that learned to program in BASIC learned many, many years ago, but really starting to introduce those to the cells themselves. And that's now advancing at what feels like light speed. Your thoughts on this area? Where is this going with logic gating?
Yeah, I think there's a very bright future for logic gating, and there's a lot of ways that cat is being skinned. I mean, it can be done transcriptionally, as what Cole Roybal and Wendell Lim did, as you mentioned, was shutting on and off selected genes. And it can be done at the level of translation or protein degradation. And so and what we don't yet know is what, I think they each have probably different applications, and all of them have promise. I mean, it depends on the time scale of how you want to control these logic gates, and the signal noise ratio of them, and if they're leaky or not, and that will depend on your choice of targets and what genes. But so this is an area where there's been very rapid progress, but unfortunately the clinical trials take a lot longer, and where the data is lagging behind, far behind what we have in mice.
Yeah, well, we'll hopefully get to that point. That is a difficult piece, the iterative importance of iterative studies in the clinic for what is still a very early field. But sticking a little bit with the concept of logic gates, right? I think a lot of the studies right now are really just focused on a single logic gate, a single switch to either turn on or turn off a CAR, or turn on or turn off something else, a cytokine, a knockout, whatever it is. But once you start engineering these cells, it doesn't feel like there necessarily needs to be a limit to how many modifications you start to make. And once you open your mind to the idea that, look, you can have multiple circuits all building into it, all feeding off of itself, it becomes really exciting and powerful, but also really, really complicated. And when you consider the fact that we're already limited, is the bottleneck in the clinic to iteratively test these things, where are we going to go with this? Where do you see the pace of advancement? How do you think we'll get to a point where we can really engineer very powerful cells that do exactly what we need them to do?
Yeah, this is where I think I have the biggest concern. We have, as you just said, I mean, the permutations and combinatorial possibilities mean that if we would do this in a series of serial experimentation and clinical trials, it'll be over a century until we find the answer of how to optimize this. It's possible AI is going to help us make some of these decisions, and I'm hopeful for that. I'm engaged with the FDA, with Alex Marson, Tony Ribas, and others, Marcella Maus now, where we actually are proposing maybe we could do screens in humans of multiple gene edits and logic circuits to find what works best, and sort of a phase zero like trial design.
Mm-hmm.
The reason I think that would be very useful is, in the mouse, tumors are just not nearly as complicated as humans, and the mouse immune system has many different species specific effects from humans, and so that some things that screen out as being useful in mice aren't working so well in humans and vice versa. So I'm hoping we could accelerate the pace of clinical discovery by actually doing phase zero like screens of these cells to find which combinations of circuits built in, edits of knockdown or knock-in of genes would be optimal.
So that seems like a really important opportunity because I just don't, then it could be 50 years if at the current pace to try to iterate around everything we need to test. AI certainly seems poised to change most of the world around us. So that will change very quickly. The concept of AI being able to start to do experiments essentially in silico, um, do you feel like we're going to be there in 3 years, 5 years, 10 years in this field? Like, how much do we need to feed it before it can really start to help us in the way of sorting out complex circuits here?
Yeah, that's an unknown. I mean, unfortunately, most of when you look at actual clinical trial results right now, they're coming out of China.
Yes.
And we're lagging behind here. We're not lagging behind in our basic research, but the clinical data coming out right now is what's going to be limiting to trained systems and and really get the benefit of artificial intelligence. So I'm hoping we can accelerate the time of initiation of just discovery trials to test what the optimal modifications of cells are. This is different than looking for efficacy. We have now great technology with biopsying, which can measure both on spatial biology and other omics and get answers with small numbers of cells as to what either promotes and makes a cell resistant to exhaustion or promotes trafficking and so on. So that is what we need to do, I think, because we have this huge menu of genes that are in the list that have been identified, but it's impossible right now to do propose modifying 100 different genes at once.
Yeah.
The way the clinical trial system is in the US.
Well, there's no doubt that China seems to have made a real commitment to try to be leaders in this space, and they've created very different systems to be able to test clinical cell therapy and try to get that iteration happening. It'll be interesting to see how the West, evolves in this area. Um, what about, short of phase zero type trials, accelerating human iteration, AI, what about organoids? How far can we get in using real human tumors, real human immune cells, but being able to create that system in a dish or an organoid-like tissue sample outside of an actual patient?
Well, there's real progress there in um, with patient-derived PDX and organoids. And so this is really critical. I mean, if we're studying a cell line in ATCC that's been there for 40 years, which unfortunately we often do, those tumor lines, they're not representative of the tumor as it comes out of a patient. And so we need to have that happen. It's actually logistically difficult to do this, takes a lot of infrastructure, but that's an investment I think we need to make that will enable discovery preclinical in a way that then can improve the what we actually take into the clinic.
Yep. Okay. Well, I'm going to shift gears here for a second, Carl. There's so much excitement in terms of, particularly in terms of technology and engineering around the synthetic immune field and what we're learning biologically to support it. But realistically, some of the most stunning data coming out of cell therapy actually has happened outside of oncology and has spurred tremendous interest in fields like autoimmunity. And I'm speaking specifically about early cell therapy experiments in diseases like lupus, right? So systemic lupus erythematosus is a really difficult disease. We've struggled to treat it clinically for years, and then suddenly we're seeing just incredible results there. Your thoughts here?
Yeah, well, I have to admit I was skeptical. The first patient, was a case report. And in the New England Journal of Medicine, it was a 20-year-old Vietnamese woman who had refractory lupus nephritis. And she was treated with the CAR that we made in my lab, an academic version of Kymriah, CD19 CAR, and she's now just passed 3 years in a drug-free remission. It's just incredible. And she only had B-cell aplasia for about 2 or 3 months, and she's back in graduate school now. So, and that was work led by George Schett and Andreas Mackinson at Erlangen on an academic trial. And they then published 5 cases in Nature Medicine, all of them responding. And then at ASH this December, they reported that they had 15 patients with 100% response rate. Now, so that's really astonishing. I think it's the only way, we would never have known for preclinical studies. Lupus has been sort of a graveyard of drug development where a lot of things work in mice, and then in humans, it's very rare to have a positive trial. So of course, this needs to be proven, but it's now been replicated by other groups, so that they're seeing responses in not just lupus, but other diseases like scleroderma, myasthenia gravis, and so on. And so this is, when we talked back in 2020, no one would have thought this. This is I think what's going to happen is that these outside of oncology areas like autoimmunity, regenerative medicine, are going to draft behind what the oncology field has established. So cell therapy, infrastructure, delivery, logistics, et cetera. And there are many more patients with autoimmunity than there are cancer. And so I think this is, over the last year, the biggest single thing that's changed in the landscape is non-malignant approaches with engineered cell therapies.
So what do you think is happening? Are we getting a reset of these patients' immune systems? Are we knocking out a malignant, clone of an immune cell? What do you think is happening?
Well, I think, we don't have all the data yet, but there had been data that with high dose chemotherapy and autologous stem cell transplants, that remissions do occur. But they were always too toxic to do that routinely, and so they look more like in many cases anecdotal. And like, for instance, if someone got a cancer and also had autoimmune disease, so then they were able to justify the high dose chemotherapy, and then they noticed that the autoimmune disease would go in remission. So now what I think we have is probably, it's what you said, it's a reset of the immune system. And then the pathogenic antibodies, in the case of lupus, well-known double-stranded DNA antibodies, the titer to those have gone to 0. And the titers, there's another disease called synthetase deficiency, where it leads to a myositis, and literally people have gone from wheelchairs to walking with a single infusion of CD19 CAR-T cells. So it's been an astonishing change, and I think rheumatologists now are learning from oncologists.
Well, it'll be really particularly interesting, as I think, if we think about, are you knocking out malignant clones versus resetting the immune system? Because I think it could change our approach and how broad this is, right? So where I'm going with this is, as you think about synthetic immune approaches here, is this really a sledgehammer for the disease? Meaning, yeah, we're getting these amazing results because we're knocking out a large population of cells, versus, no, we can refine this. This is just the beginning, right? We're using a sledgehammer right now to knock out all your B cells or plasma cells, but where the field's really going is to be now start to build scalpels where you can very specifically knock out a particular clone, whether that's a malignant clone or not, or knock out a specific cell that's really, I don't know, causal in the pathology.
Yeah, totally. I think it will be, so far it's less toxic than it is with oncology patients. So there's been very minimal cytokine release syndrome, but clearly in a non-malignant disease, it needs to have a very high therapeutic index.
Yep.
And so far, it looks like you don't need long-term B-cell aplasia to achieve, benefit, at least in the reported cases. So it may mean that we don't need long-term CAR T cell persistence, which does correlate with efficacy in leukemia.
Yep.
So it may be that, this may be a big opening for bispecific T cell engagers or
Right.
other ways to make allogeneic off-the-shelf CAR T cells, for instance,
That's
or
right.
NK cells even.
Yep. You mentioned regenerative medicine. I don't think many people think about regenerative medicine in this particular area of cancer immunotherapy and synthetic immunity. But as we think about cell therapy, it does seem like a really amazing and potentially untapped use for engineered cells.
Oh, totally. So, the fact that we don't need them to persist long, at least in this initial day, is really bodes well for this. And I think, when I looked about a month ago on clinicaltrials.gov, there were 34 different trials, both with pharma, biotech, and academics, using CAR T cells for autoimmunity.
Yep. So we all look forward to seeing where that goes. All right. I'd like to move. We're going to close out our section and move into Q&A. But before doing that, I thought maybe, Carl, you and I could give a quick closing. And one of the things that I would ask you to say in your quick closing is, if we're looking at 2030 or 2040, and CAR-T has either, or cell therapy has either cured cancer or flailed at curing solid tumors. Can you at least say what you think would have led it to be successful to cure cancer, and what would have happened if it didn't cure cancer?
Well, sure. So the big issue is in the space of oncology or solid tumors, as we've discussed. During this last year, we saw two major publications with CAR T cells, but in rare pediatric tumors. A group from Stanford showing that diffuse midline gliomas could have benefit with repeated infusions that were locally instilled into the intracranial by Ommaya reservoirs. And then a group in Italy showed that pediatric refractory neuroblastoma could enter remission with CAR T cells. And that hasn't happened reproducibly yet with solid tumors in adults. So, but I think that will happen by 2030. In some tumors, there will be enough progress that we'll see that. So the issue then becomes, will we be able to get away from autologous cells and the complex logistics of that to either, off-the-shelf cells or, in vivo manufacturing of cells? And I think that's going to be required to meet the scale of demand that we're going to see with that success.
Okay. From my standpoint, I guess if we have been successful in 2030 or 2040, we will have solved the issues around overstimulation, and gotten better engagement of endogenous immunity, which feels still very lacking in a lot of synthetic immune approaches, at least good evidence for strong engagement of endogenous immunity. And I think success probably also includes things like in vivo CAR-T, because I think that that's an approach where we will be able to overcome enormous challenges just by creating something that's logistically easier and can be redosed to recreate biology, even if you can't make it sustained with a single dose. I think that if we've failed because we haven't overcome that problem, and we can't dissociate the type of burst activity we see with synthetic immunity from the ability to really get good long-term functional biology out of the cells or the engagers that we're giving. So it'll be really interesting to see where that goes. All right, let's go ahead and move on to the final, in the final 10 minutes, we'll go into Q&A. I'm going to start us off with a question so that I can then start taking a look at all the questions that are on the board. But Carl, maybe a first question to you. So endogenous immune approaches have struggled with the next generation of checkpoint inhibitors, but where the advances have come is actually pushing current checkpoints into earlier disease. So whether it's MRD space or the adjuvant setting, and we're starting to see activity for things like vaccines in early studies that maybe we didn't see in the metastatic setting, suggesting that the immune context may be very different in that earlier space. Do you see that as a place where cell therapy and synthetic immune T-cell engagers will go? I haven't seen those trials, but it seems like an incredible opportunity that is still waiting the field.
Yeah, I think that's a critical thing. So what you're referring to, Dan, I think is a paper in Nature last year from Balachandran group at Memorial Sloan Kettering and Genentech and BioNTech, where they, in early stage pancreatic cancer, and this is a very specific subset of patients, those that had resectable tumors, then in a neoadjuvant setting could get vaccines against up to 15 different neoantigens. So they were able to, sequence the tumor and then make RNA lipid nanoparticles to make a vaccine that would be given to the patient, go into dendritic cells, and then prime an immune response that had not yet occurred, but against their tumor-specific neoantigens. And so it was an early stage trial, but it looked very promising in that in the pancreatic cancer patients who responded to the vaccine, they basically did not relapse. Whereas if their immune system didn't respond to the vaccine, they had the usual course of pancreatic cancer, which is relapse and early progression. So if this is replicated in ongoing later stage studies, we'll soon know that in a year or so. That means, I think, that first of all, vaccine strategies have failed for many years because they were given late, as you mentioned, and also that that's where we need to take, bispecific engagers and engineered and synthetically enhanced cell therapies. It may be much more effective before the much more toxic tumor microenvironment is established in late stage cancer.
Yeah. It seems like an amazing opportunity. I'd be very curious to see how synthetic immune approaches work without that big bolus of disease to start with as well, right? So maybe we see a lot less CRS when there's not a mass of cells just sitting there. All right, so questions from the board. So the first one I'm going to pull off here is a really important one for the field. So the question is lymphodepleting chemotherapy a critical part of these synthetic immune approaches, whether it's in cancer or in autoimmune disease? So, obviously the current technologies generally incorporate lymphodepleting chemotherapy. A lot of the advanced approaches coming seek to remove that and give much greater flexibility and tolerability for patients. But as we do that, are we actually going to remove a critical component of cell therapy?
Yeah, well, this needs to be experimentally determined. I mean, there are a number of approaches in the lab to where the cells are being engineered so that they then don't require the conditioning chemotherapy, which is usually, but not always, fludarabine and Cytoxan. I think those are really going to be essential to work that out for the widespread use of this in autoimmunity. In cancer, the patients are already exposed to chemotherapy, and it's not such a big threshold, whereas in most rheumatology patients have not had alkylating chemotherapy. So this is going to be worked out, I think, in the next 5 years. This is something I'm quite confident that we'll have both approaches using cell therapies that require lymphodepletion and others that don't.
But the ones that don't, you don't think we're going to be losing some critical aspect of the therapy? Meaning,
no, I think because I think there are ways to make cells where they can get their own niche. And there's a company already for myasthenia gravis that's reported phase 2 trials where they gave repeated infusions of BCMA CAR-T cells and no lymphodepletion, and have clinical measures of efficacy. So there, it's not a single infusion, but the repeated infusion seemed to remove the need for the lymphodepletion.
Okay. I'm going to combine a couple of questions here for the interest of time. So there's some interesting questions around solid tumors and some of the more advanced thinking around the biology of solid tumors that can make them very difficult to treat. So the question is, as we think about synthetic immunity in solid tumors, what is the opportunity to combine it with things that affect or deplete tumor stroma? And what are the opportunities that might affect the metabolic derangements that we can see in the tumor microenvironment? So can we address things like mitochondrial behavior or other ways to change the metabolism of these cells we've engineered?
Yeah, so I mean, a big issue is in many solid tumors, the cells just don't traffic in, to these extremely cold tumors, and has been seen repeatedly in advanced pancreatic cancer and many other solid tumors. And we recently did work with Ellen Pure's lab where we combined two kinds of cell therapy, a CAR that targeted the collagen scar in pancreatic cancer. So this is in mouse models, but if we gave a CAR targeting the tumor in the mouse pancreatic cancer, nothing happened basically. It did, the cold tumor stayed cold. But if we gave a CAR targeting fibroblast activation protein, which is on the scar forming cells, and then a week later gave the same CAR T cells, but against a tumor that didn't work by themselves. Now we had a clinical eradication of the tumor in mice. So I think that looks really promising as a way to go to remodel the tumor microenvironment. And I think, other approaches could do this, potentially giving different kinds of engineered viruses, oncolytic viruses and so on, that could reprogram the tumor and then allow antibody access or protein and cell therapies that otherwise would not work.
Yeah, so I mean, it feels like a particularly important approach in the excluded tumors, so your pancreatics and colon cancers in particular. So you are working on building these types of approaches in. What about the metabolic side?
Yeah, well, I mean, there's both lots of work in immunometabolism now. It's become an entire field. And part of it is, I mean, it turns out different lymphocyte subsets function better with different fuel sources.
Yep.
And it's very different for a regulatory T cell that shuts down the tumor immune response versus an effector T cell. So there's a lot of work on that, on giving fuel to T cells when they go into the toxic tumor microenvironment. And then there's changing the metabolism of the tumor itself, the so-called Warburg effect, where they have aerobic glycolysis, and that then makes a steel for metabolites and so on that the immune cells need to flourish. So a lot of approaches here with the synthetic immunity to improve and the metabolic features in the toxic tumor microenvironment.
But do you go about changing that, or do you engineer it in? Like, how difficult is it to just engineer it in so that the T cells you want to have functional are able to continue to maintain that functional state in a metabolically hostile environment?
Yeah, there's a number of approaches, both genetic to change, for instance, the Krebs cycle ability of a T cell versus pure oxidative and versus the anaerobic metabolism. But we even have some really exciting data, and there's others, with just by enhancing the cell culture of cells with higher concentrations of L-arginine. Normal cell culture media is 1 millimolar arginine, and if you increase it to 5 millimolar, there's genetic reprogramming of cells, and they work much better in mice for months. After so some of this metabolism changes can happen with this cell culture media and so on, and others is going to happen, I think, in vivo in patients.
Well, I wonder if you can give your cells a little backpack full of fuel.
Yeah.
And when you feed them well, during production, I wonder if some of that is really just increasing stores of metabolites that they would then be able to carry into tumors. But it's an incredibly interesting space, I think, for the field to advance in. We're just about out of time here. On last closing question. I myself, I'm very T-cell centric in terms of the approach to synthetic immunity, but obviously there are a lot of other ways, whether it's NK cells, macrophages, gamma deltas, that are ongoing. Your thoughts, you commented a little bit on this earlier, your thoughts on expanding beyond T cells to either use different cell types as synthetic immunity or a community of cells as synthetic immunity?
Yeah, I think it's going to depend on the indication. There's going to be many kinds of cell, both natural, for instance, natural NK cells, cord blood derived, or iPSC derived cells of NK cells, NKT cell subsets, et cetera, and macrophage. And we'll also see, just this last year, FDA approval of stem cell engineering to treat hemoglobinopathies. That's a huge thing this last year. And that's just the beginning, I think, of what we'll see, stem cell engineering.
Well, thank you so much, Carl. Thank you, Kate and IO360 for making this happen.
Dr. Chen, Dr. June, thank you again for an incredible discussion today. Again, this webcast was brought to you by Immuno-oncology 360. As a reminder, we're celebrating our 10th annual IO360 Summit, February 27th through 29th in New York City. For more information about IO360, you can visit io360summit.com. Thanks for joining us.